A short introduction to HistoFlow. HistoFlow is a software that enables to apply and to train deep learning models for cell segmentation and analysis. I will start with showing you how to apply a deep learning model to a histology image. The first step always is to create a view, and then we can drag and drop the new image onto the application window, like this. Now we have our histology image here, and the software works best with fluorescence microscopy images, but you can train a model for whatever image you like. The next step is to add a CNN inference block. We will then connect the image to all the input channels because the model is trained for grayscale images. The next step would be to connect a model. You could connect a custom trained model here. In this case, we will use the default model and leave this node unconnected. And now at last you just click on run. The image is sent to the server, and as a result we will get back the output image of the neural network and a dataset of all cell centers. We can visualize the cell centers here, so you can see that each cell is marked with a green dot. The last step to get a real instant segmentation of all cells is to add a region grow block, and we will now apply a region grow algorithm to the image and the cell centers. We click on run and we will see that the algorithm found the boundaries of all cells by applying a region grow algorithm starting from the centers of the cells to the edge of this mask that the neural network returned. So this is everything you need to segment cells in the software using deep learning. Now I want to show you how you could train such a model. So on the GitHub repository you can find example projects. You can just download it in this case, and we'll download train using artificial data. I already did this, and you can now just drag and drop the file onto the application window like this. It will ask you whether you want to import the project. You just click open, and now you have your example project here. I have to load the image as the first step, the image that will be used as example data. I already downloaded it and can select it here. Okay. So now we can see here our image that we will use as an example image for the new trained model. The next step is to annotate some example cells. These will be used to get the minimum and maximum cell size. 3 cells are already annotated. I will annotate one more cell. We have to check whether interactive watershed is selected here. It is. And now we can just click and hold and drag the mouse to annotate a cell. And now that we have annotated some cells, we can use them as examples for new randomly generated cells. So the next step is to just click on generate here. This block will generate new random cells and fill them into this dataset. So this is done now. The next step is to render those random cells. So those random cells here in this dataset block are not visualized anywhere yet. They are just data, and we'll now render them as a new DEPI image. For this I will first hide our original example image and our segmentations. And now I can refresh here and see a preview of our DEPI image. I click save and a new DEPI image is generated. We will use, you can see it here at the top, we will now use this artificial DEPI image as the input to our training data. As the target, we want to use a mask for the first channel. We can select mask here and save this as an image 2. We connect it to the first target channel, and then we'll also generate a center channel like this, and we'll use the centers of the nuclei as the second target channel. Here you can see what the model will do later. It will learn to convert this DEPI image into this ground truth image, and we'll use this to segment the cells later. Here we can now generate patches from this data. So the input images will be randomly cropped, and patches will be generated and augmented by noise and brightness changes and some other things like rotations. I will generate 50 patches for this demonstration. Usually would create like 2000 patches. So I click generate all, select a folder to store them, and select a file name. Those 50 images are rendered pretty quickly. It will take a little bit longer for 2000 images. So now we have the training data here, and we can connect the training data to the CNN training block. And usually you would have to repeat all the steps a second time to generate validation data with fewer patches. For this demonstration, I will use or created training data for both training and validation data. Of course, the model will overfit with this, so this is only for demonstration. We can enter a name for the new model and select 2 epochs. And we could also connect a base model to use transfer learning to use an already existing model as our initial weights and retrain them with other training data or fine-tune them. So, but our last step here is to click run. It will upload the data to the server, and the model will be now trained. This is the first and second epoch. We will now validate it, and after the validation is done, we can use the model. So now this is how a model is trained. This was just trained in real time. Using more patches, of course, will be longer, but it will still be under about 20 minutes. And now we could use this CNN model to segment cells like showed earlier. Okay, so I hope this is a good insight into what HistoFlow is able to do. You can see how analysis of their cells and training a model for classification and classify single cells is done by having a look into the other example projects, and more information is also found in the GitHub README. Note that you have to set up a server for all this to work. Beside that, you can just use the Windows and macOS binaries. Thank you for your attention, and yeah, I hope this is a useful software for you.